Good morning. I am Rho Mi-na from Hanyang University. Thank you for inviting me to this important forum. The topic that I will cover today is deep learning approaches for deciphering composition and functional roles of the ocean microbiome. So today I will explain how we applied AI and deep learning on studying ocean microbiome. As you well know, microbiome can be Microbiome consists of microbiota and theater of activity, so we don't have two cultures, so those are uncultured. So we need to comprehensively study the environment as well as their theater of activity. So first we need to know which kind of microbe exists in certain environment and how they function in those certain environment. So the problem here is that when we transfer that genetic information into data, first we had expected that if we have the genetic information, then we can convert those data into the true microbe or so. But it was difficult to recover the genetic sequence of, or the genetic information, fragmented genetic information that we had. So we changed the study research approach. So instead of recovering the fragmented genetic information, we focus on the functional role of the microbe. So the microbiome research is gaining a lot of attention, especially in the human microbiome research. But the big R&D project is on ocean microbiome. As you can see, the consortium was formed in early 2000. And 20 years have passed, and a lot of big consortiums were kicked off to study ocean microbiome. Especially in ocean microbiome, we see the relationship between ground and ocean and the different functionality between ocean and ground. So in many areas, we see a lot of proteobacteria in vast area of the ocean. And the functionality, especially the energy functionality of the microbe. So in the past, we tried to reproduce each microbe functionality, but instead now we focus on certain environments, so whether certain specific feature is enhancing the microbe. So as you can see on the slide, we study coastal area of Korea. We studied Jeju Island and South Sea. We saw seawater sediment and seafloor sediment to study the differences in functionality, and there was a clear differences. And through the pathway, functionality pathway, we saw the distribution of enzyme within the microbe. So this is the research that we have conducted in our lab. So environment microbiome, especially ocean microbiome, the importance it has is because of the relationship it has with humans. So recently, the One Health approach, you can confirm that in the recent research. The ARG gene is getting a lot of attention because it can be transmitted to human. So in order to study that, we need to see environment and the product that comes from those environment. And if they are included in that, then it can directly transfer to human. And according to one research, by 2050, the ARG gene can contribute to death of a human in a large scale. So that's why we need to study this field. The previous microbiome research was more of task-specific research. But nowadays, it has changed. Through data production, as we can use the AI technology, we can use the AI algorithm model to understand the problem and extract a solution based on that. So one of the representative example is what you see on the slide. The three renowned experts in AI, they wrote a review paper in 2015 while explaining the deep learning. And they pinpointed drug discovery and genomics as a promising field out of AI. The reason behind this is because the chemical compound and genomics DNA sequence data size is quite big in the biometrics industry. If the vast amount of data can be learned through AI, then the result will be very promising. So in the past, we only focus on task-specific research, but now we can use a deep learning algorithm for a more generalized solution. So for example, as you can see from the personalized medicine case, you can see the effect between or interaction between the medicine and the specific human. So likewise, the focus is on how we can optimize the model to suit the genome. The most used algorithm, deep learning algorithm, is large language model. As mentioned in the past, large language model was based on rule-based model, task-specific rule-based. So it was a database, but nowadays it is more of neural network based. So it has a vast amounts of parameters, and research nowadays focus on this neural network based large language model.
It seems that language models can also be highly optimized for our biological sequences. The reason for this is that elements like sentences and words in language models can be applied directly to genomic data, where units like genes, amino acids, and nucleotides, as well as their relationships, are reflected. These motifs and coevolutionary mechanisms of interactions allow the model to discover various rules. For instance, AlphaFold, which is currently very popular, was able to capture the relationships between distant amino acids by using the biological mechanism of coevolution. Secondly, these language models, through self-supervised learning rather than task-specific learning, can effectively capture general features as seen in images. For example, a task like recognizing that an image has been rotated 90 degrees helps the model learn features of the image. In language, masking a word in a sentence and then predicting the masked word helps the model understand the sentence's meaning. Similarly, in genomic data, a masked segment of the genome can be used as a task for the model to learn the features of DNA, in a way very similar to language models. Even though these fragments may seem meaningless at first glance, certain parts of the DNA play crucial roles in evolutionary conservation. Through the model's learning process, these unknown elements are stored as parameter variables. By utilizing this approach, we have been able to address many issues in microbiome research, such as problems with taxonomic profiling and functionality. Now, I'll show you 3 brief examples. The first one involves taxonomic classification, where, as I showed earlier, we developed a model using sequence fragments obtained from environmental samples.
Accurately allocate the taxa of that fragment and can know which virus or strain that taxa is coming from. And ultimately, the decision can be made. The biggest impact out of this, as mentioned before, is that in the past, the biological sequence was based on homology, but now, because in the past method only can study the closely related protein or taxa. But if we use the learning model, then we can see the entire feature of the microbe. So we can study the remotely homologous genes and do taxa prediction. So one of the representative case is this one. As you can see, the recall rate of our study has 74% of accuracy, but compared to the past method, it only had 20% accuracy in the prediction. So the recent big data-based approach can do better generalization. So the data points that the algorithm didn't study can be called as reads. If the interrelationship between the read is not identified, but if the algorithm has been trained using 4 viruses, for example, coronavirus, if we put in the COVID sample in it, then the algorithm can tell that there is a similarity between the sample and beta COVID virus. So in function, as you can see on the slide, the interaction between the two is studied. And in the past, the accuracy was only 30 to 40%. If so, the similarity is only 30 to 40%. That leads to false positive results. But now, through training the algorithm, then the percentage increased. The similarity percentage increased. Likewise, you can see identify convolution and attention-based sequence. So function conservation and secondary structure are well trained using this model. So the structure you see on the slide, the structure is different, but in the past, the result was quite vague. But using convolution and attention, when we train the data, then the feature is well structured. And as you can see on the right side, the similar one has similar vectors. And third, using genome exchange, we studied the genome exchange. And one of the main fields is ARG, antibiotic resistance gene. And there are a lot of materials that is carried by ARG. And we need to study the comprehensive structure of those genes because they carry different materials. So we mapped that ARG gene and used the algorithm to understand. So this is the true sample case, the case we use the sample, and this is the targeted ARG. But the ARG can exist remotely from the sample gene. If we do target research, it may seem that the target research is done well. So this is the case where we used the previous method, but now we have modeled it better. This is false positive results. So homology-based tool, the conventional tool, there were some cases that it correctly answered it or not, but deep learning model can understand general feature and specific features very well. This means that the correctness of predictivity is quite high. So today I covered how deep learning can be used to understand microbiotic data. And multiple representation can be recovered through deep learning. So if there's a lot of data, for example, genomics or chemical data, then deep learning has high predictability or accurate rate of predictability. If used self-supervised learning and the training, the generalization and specification are done well. And that contribute to well understanding the genomic sequence. That's why we use the deep learning in a lot of genome fields. I think this is very promising technology. Currently, this research is done by AI Lab of Hanyang University and a lot of R&D projects that is backed by MOF. Thank you for listening.